Hello everyone, so I am Chloe Geagan. Like mentioned, I'm based up in Newcastle. The eagle-eyed among you will also notice that I work with Rory Conn and Janet Hoskin and lots of other people in this room on the psychosocial project in Duchenne as well. So today I'm here to speak about the BIND project. This is just a bit of an overview. Full disclosure, analysis is ongoing. So there's not a lot of data on these slides. This is just to give you a bit of an idea of what's been happening from within the BIND project. It mentions there the BIND consortium. So just to let you know, the BIND project is a European study. There's 7 different countries involved. 6 different countries across 7 different centres. And you'll see there we've got Denmark, France, Netherlands, UK, Italy, and Spain. So what does BIND actually stand for? Interestingly, Volker said this morning about not using the word dystrophinopathies, but actually the study, BIND, stands for brain involvement in dystrophinopathies. And what we already know is that some individuals with Duchenne muscular dystrophy and also Becker muscular dystrophy, so the dystrophinopathies, have issues with cognition, so sometimes with learning, with understanding, behavior, or both, okay? So in the simplest terms, some of the loose aims from the BIND study were, why do these neurobehavioral symptoms vary? So basically, why do some individuals with Duchenne struggle more than other individuals? How do we actually check for these difficulties? And if we do find some difficulties, what can we actually do about it? Okay. So the overall aim is actually just exploring the learning and behavior within Duchenne muscular dystrophy and Becker muscular dystrophy. So what we know already, you have heard this a lot from yesterday and even this morning, that Duchenne is not just a muscle disease. We know that dystrophin, so the dystrophin protein, is found in both the muscle and the brain. And depending on where the genetic mutation occurs, different forms of brain dystrophin can be affected. So that's kind of within the BIND study, a focus is quite a lot on the genetic mutation and seeing whether there are certain patterns or profiles that might come about based on where the mutation actually occurs in the gene. Some people might have already seen this slide. I can't quite remember if Janet used it yesterday in her talk or Rory used it yesterday, but Jos Hendrickson from the Netherlands speaks a lot about the Big 10 in Duchenne. So when we're kind of thinking about that, we're looking at some of the areas that we know for some individuals can be difficult. So we look at cognition, so our standard intelligence test, basically our IQ. We look at things like work and memory, which is exactly what's happening today, where someone's up on a podium, we're talking at you, you're taking in the information. Some people are taking notes, which is probably helping with your work and memory if you want to ask a question or take things on, brain-wise. And then we've got other kind of areas like executive functions, so planning, organizing, being able to regulate our emotions, being able to regulate our behavior. They're really, really, really important life skills that do take quite a long time to develop when we think about executive functions. So the main example being teenagers, you know, a lot of the times kind of exploring our planning and organization versus when you're an adult. So these are all developmental aspects from a cognitive point of view. Then we look at areas like academics. So we heard from Saul and Benjamin yesterday around actually some difficulties from a dyslexia or reading difficulties, because not everyone might have a diagnosis of dyslexia, but also dyscalculia, so difficulties with numeracy, numbers, maths. Again, for some individuals with Duchenne can be really, really tricky. Then we look at things like our more emotional, psychological side of things. So stress, anxiety. We know a lot of individuals with Duchenne might present as more anxious for lots of different reasons. And again, we talk about the wider biopsychosocial model, but again, from the BIND project, we're looking at it from a kind of genetic mutation point of view of, are you more vulnerable or are you more likely to experience anxiety or mood difficulties depending on where your mutation is? And then we have behavior. So again, different words are used in different countries and different places, but we might think about things like neurodevelopmental conditions such as autism, ADHD, and then we also have, for some individuals, OCD or OCD-like traits, obsessive compulsive disorder, just in case. But real life is not so simple. So my job would be a lot easier as a clinical psychologist who works in neuropsychology, so that link between brain and behavior, if everything was nice and neat in these little circles. However, in real life, there tends to be a massive overlap, okay? a lot of the times, anyone who's been through the process of going to CAMS or SIPs for an autism assessment, ADHD assessment, etc., basically what you're doing is you're ticking a certain number of boxes to be able to get a certain diagnosis. What we know from Duchenne and from Becker as well is that it's not that simple. Lots of individuals might have certain traits of a certain condition, but might not meet a certain diagnostic threshold. So it's just to highlight that when we're looking at these kind of things, it's never a clear, simple answer of, oh, someone has difficulties with their memory. There might be lots of other things going on. So were they feeling particularly stressed that day that made it harder for them to engage in the assessment? Are there some, just there's lots of different factors that can combine to lead us to have lots of different areas like this. So from the bind point of view, there were a number of different what we call working packages, okay? Just for ease, we split them into the kind of preclinical studies and then the clinical studies for today. I will not even pretend that I understand mice models. There's a lot more, there's many experts in this room, likewise from a gene point of view, but basically the preclinical studies were the ones using our lovely mice participants, looking at the way different types of dystrophin affects different brain areas, okay? Trying to look at whether they could find consistent signs or symptoms within mice models depending on which mutation the mice had. So the way they did this was they also used some of the tasks that we used from the clinical studies, so like, finding your way around a maze, different kinds of social behavior, how mice were interacting to basically study the behavioral aspects of having different mutations. And then also thinking about, can behavior be improved in mice with genetic therapies focusing on treating the brain dystrophin deficiency, okay? You will notice I am slightly biased in that as long as we're going to spend on the preclinical studies. And I'm going to swap to the clinical. So again, the main aim of the clinical studies, we're looking at how do we understand different mutations in Duchenne and Becker, how they affect individuals, and how severe these effects are, okay? So the way we did this, I'm very aware there's some individuals in this room who actually took part in the BIND study, so this might sound a bit familiar, but basically part one of the study looked at doing, some online questionnaires. So it was usually parents or caregivers who answered some questions around behavior, mental health, a little bit on history of developmental milestones, all the stuff that you get asked in clinical sessions anyway. Part two is the bit that I love, and this is the bit I was mostly involved in because it involves sitting down in a room with a young person doing a neuropsychology assessment. So again, we're doing lots of different games and puzzles. On the top right-hand corner there, you can see just an example of one of the tasks that we use, where it's your traditional IQ tests. We did some memory games, some attention ones, etc. It was a very long process. You're looking at 2, 3, 4 hours, depending on the young person in front of you. But that's the one where it gives us some of our numbers of what someone's cognitive profile is. And then there's part three, which was introduced a bit later into the BIND project, which was actually developing a screening tool. So again, this is still in progress at the moment, and they're still gathering some data from this side. But we might have heard a little bit of actually how do you screen for some of these difficulties? How do we support clinicians and families to actually pick up if there's some potential difficulties early on? So that was kind of what one of the goals from the BIND project was to develop a tool that would help clinicians to kind of assess for this alongside, asking questions around muscle and breathing and cardiac side of things as well. Also included within the clinical studies was the brain imaging bit, so taking part in a brain MRI scan. Again, quite a long process. Very interesting seeing people's reactions to whether you wanted to do the MRI scan or not. Some guys were like, yes, 100%. Other guys were like, no, you're not going to get me to go into an MRI, which is absolutely fine. But basically, from the brain imaging side of things, we were looking at the actual structure of the brain. So what does the brain look like? Volume, white matter, grey matter, all the brain-related side of things. But also the function. So how is it working? Okay. So that was kind of like lower right-hand image of you do a task or something's happening in the scanner, which bits of the brain are basically lighting up when you're doing your scan. And one of the other goals then is to actually, can we link some of the neuropsychology, so that, the learning assessment side of things with the brain scans, and just see if we can find some specific profiles within Duchenne. So how did it go? Like I mentioned at the start, I can't be super detailed on data, but please catch me afterwards if anyone has any questions. So within Bind, we recruited, like I mentioned, Duchenne and Becker. From the Duchenne side of things, it was 5 to 17-year-olds. So from part one, we had the kind of shorter questionnaires. We had 277 parents and carers complete the questionnaires across all of our sites. 238 participants completed a much longer version of a questionnaire, which is a bit more diagnostic based, like looking a bit more into whether we could possibly do something like identify anxiety or ADHD based on some questions. So it wasn't a diagnostic assessment. It wasn't a case of if you took part in Bind that you then, if you met criteria for autism, you would get that diagnosis. It was just trying to see if we can improve the clinical tools that we have, because again, there's no biological marker for things like ADHD or autism. We do very much rely on interviews, observations, clinical reports, etc. So it's just trying to see how sensitive and how useful the measures are that we use are at picking up some of these difficulties. And then likewise from part two, we had 253 families take part in the more one-to-one assessment with a psychologist or a research assistant. Not everyone completed everything, because I don't know if how many people in the room have had the joy of trying to assess or work with a 5-year-old for 2 hours versus a teenager. So again, there was within participant kind of differences as well of how much we could actually complete within the time frame we had. So again, I'm very aware it's getting recorded. I do have, please don't take a copy of the image. The main reason I say that is it's probably going to change. This is very much preliminary. Like I said, hopefully this time next year, we would have some more kind of set data for you to kind of be able to use and show people and show schools in particular. And I know Janet and Nick are doing a session, like a breakout session around education and stuff. This is where this kind of stuff is really, really important, okay? So we'll see here, again, anything in blue is within what we call the average range. So basically what we would expect for someone when compared to other young people their age. So not other young people with Duchenne or other health conditions, literally from the normal population, basically. So you see here from these are general cognitive abilities, basically the IQ side of things. Majority of young people who took part in our study were performing within the average range. You'll see around 35% were in what we call the borderline. So anything in yellow is at borderline, so probably at risk for some level of difficulties or likely to have some level of difficulties. And then around 20% were performing in the very low range or the extremely low range, which would suggest that we've got quite a significant level of difficulty from a learning point of view. We then looked at things like memory, but specifically when in Bind, we just looked at verbal memory. So how well someone was able to do when we gave them some words to remember. You had a chance to learn it across, a few different tasks. It is quite boring. So this is, I'll be completely honest on it. So again, this is where we're thinking of, when you see the image with all the overlapping circles and stuff like that, you're looking at memory, but if someone's really bored and you're losing your attention, you're not going to get the best out of someone from it. So I think take it with a pinch of salt, but we do see for both Duchenne and Becker an auditory memory bit that actually this was an area of difficulty for a lot of the participants in our in the Bind study, and just thinking about actually real life what the impacts of that might be, particularly from a classroom point of view. The next one was the executive functioning, so that there's so many different tasks that you can use for executive functioning. We just looked a little bit at planning, a nice, simple, straightforward one that actually I think most people kind of enjoyed a bit more. Again, not particularly concerning, but some areas of difficulties there. And then we've got our reading and our maths, okay? With the reading, just to make it clear, so a majority of people scored in the average range. Just wanted to highlight that was on a really simple word reading task where you basically were just reading out some words. It didn't involve reading a longer passage of text and then telling me what happened in the story, etc., etc. It was purely just read these words out loud, okay? So again, if we're getting our clinical hat on, and something we'll explore a bit further on, is if you were to do something more complex from a reading point of view or literacy point of view, would you see some more areas of difficulty there? And then likewise from numeracy and arithmetic, you'll see that this actually was flagging up as a bit more of an area of difficulty. Again, it wasn't super complex maths that we were doing. It was a bit of a speeded task. but kind of simple, like addition, subtraction side of things. We weren't being cruel and asking people to do loads of fractions or algebra or anything. It was basically how quickly can you do basic maths? And we noticed that was, again, a bit of an area of difficulty for a lot of our participants. So, in summary, we know that cognitive functioning is affected, but that doesn't mean that everyone's going to have difficulties. There does appear to be a specific cognitive profile, but again, when we say that, it doesn't mean that you get a diagnosis of Duchenne and therefore you're going to have X, Y, Z difficulties. It just means that we're noticing certain patterns are getting highlighted that do appear to be a bit more difficult within Duchenne, which we kind of know a little bit from the literature already. Within the BIND study, the verbal memory side seems to be the most problematic area, but again, we're going to look a little bit further into why that might have been the case. Again, numeracy was a bit trickier than the reading task, and also there does appear to be a significant effect depending on the mutation group. Okay, so going back to that idea of one of the key aims for BIND was if you have your deletion in XYZ exon, what does that mean for you from a learning point of view? But again, going back to the point that was raised, in the questions just beforehand, we could also have siblings taking part in BIND who both have the same genetic mutation, but they perform differently on our tasks. So again, it's not to say just because you have that specific mutation, you are then going to go and have specific difficulties in that area. So where have we got to now and what's next? So recruitment is closed. Like I mentioned, analysis is ongoing at the moment. Just to flag up, individuals who have taken part in BIND should have received feedback on how your actual child did. If you haven't got a report, please just catch me somewhere after the break and we'll try and find where that's gone for you. Anna Kolesnik from London, UCL is here. Yep, there as well. So anyone who took part from GUSH side of things, try and find Anna. If not, come and catch myself and we'll find out where that's gone to for you. There's also the BIND screener, which was the part 3 bit, which I'm just going to mention as well at the end of this brief little overview. And then there has been talk of BIND 2.0. Professor Montoni was going to do this talk originally. He probably has more details. Maybe Volker might know more. But either way, the idea is post this study, we're meant to be continuing on to actually do the next steps within the study and explore certain things in more detail. Key take-home messages. So again, you're going to be sick of hearing it, but Duchenne is not just a muscle disease. Not everyone with Duchenne will experience learning or cognitive difficulties, and there is a big overlap in clinical diagnoses or variance within the population as well, so that heterogeneity in scores. I've gone rogue and I have put in a BIND template, sorry, it's very red, but just really want to say thank you to everyone who did actually take part in the BIND study. It is really, really useful because it's an area that is growing. There is a lot more interest in the brain involvement in dystrophinopathies. And just wanted to mention, just going to put this out at the moment, if you want any further updates, that is the BIND website. Please feel free to take a picture. You still have a chance to take part in the BIND screener where they are collecting data. It's very, very short. I think it takes 5 minutes. Again, please feel free to give feedback. I realize I haven't put my email address on it, but if you've got any comments on it, because then it's at the stage where it can be changed and tweaked from a language point of view if there's certain things you're like, that just doesn't feel right. But if you do want to take part in that, go wild at some point today. And I think that's it. Yeah.
So hello, good morning. My name is Jordi Diaz Manera, and I work in Newcastle University. I'm going to talk to you about fibrosis. The title of my talk is Latest Translational Research in Fibrosis. And I think I have a difficult task because I'm talking to an audience that is not expert. You are probably tired, you're probably under-caffeinated and hungry. And the last thing before lunch. So on top of that, as Annemieke said, I have not been trained to talk to a lay audience, but I will try my best. So let's go with this. So first important thing, you have heard about fibrosis probably several times this morning, and maybe you don't know what is fibrosis. So the idea here today is to talk what is fibrosis, why fibrosis is bad for the muscle, what can we do to remove fibrosis from the muscle. So the first thing is understanding what is fibrosis. So fibrosis is basically the excessive accumulation of a scar tissue that we can have in different organs. And this is basically when the body tries to block or reduce a chronic inflammation of a chronic injury. And it occurs when the body produces too much collagen. So collagen is a component of what we call the connective tissue. And the connective tissue is important for the organs. It's like the foundation and the walls of a house. It's where all the cells display. So it is important. But under these diseases, and when we have a chronic disease, our cells start producing a lot of this connective tissue. And this is what is wrong, and this is what is bad. So this can lead to thickening and stiffening of tissues, impairing their normal function. And this is not only happening in the muscle, it can happen in many different tissues. It can happen in the lungs if you have a chronic respiratory disease. It can happen in the liver if you have a chronic liver disease, in the skin, for example, and in the muscle, which is the disease that we are interested today. So how is fibrosis developed in the muscle? So one important thing is that when we look at the muscle, a normal muscle, sometimes we think that there are only muscle fibers there, which are the cells that contract and produce the movement. But in the muscle, we have many other cells. And there are two that are relevant to this talk. One of them are what we call the satellite cells, which are cells that can proliferate and differentiate and contribute to repair the muscle. And another one is what is called the FABs, which are the fibroadipogenic progenitor cells. So you will hear about these cells a lot today. So when I have an acute injury, for example, a sports player, a football player have an acute injury of the muscle, the muscle has an incredible capacity to regenerate. And this happens because I have an injury, then the first thing that happens is that there are going to be inflammation, so inflammatory cells will arrive to the muscle. These inflammatory cells are good, are pro-regenerative. They will release a series of factors that will increase the number of FABs, that will produce this connective tissue, and then also increase the number of satellite cells that will regenerate the muscle. Okay? This is what happens when I have an acute injury. In Duchenne, there are endless cycles of injury, regeneration, injury, regeneration, injury, regeneration, because the new muscle that is produced is not normal. It lacks dystrophin. And when this happens, we enter into a different situation, which is this one on the bottom. So we have now an inflammation that is basically composed of cells that are pro-fibrotic. Okay? So they are going to release factors that are going to activate these FABs, and they are going to proliferate a lot. And when this happens, they are going to differentiate into fibrotic cells and produce this amount, excessive amount of extra of connective tissue, but they can also differentiate into adipocytes, and they will produce the fat. And this connective tissue and fat will start substituting and replacing the muscle fibers that are dying. So this is how it looks like. So if we start from a healthy muscle, in early stages, we can have a bit of this pink thing here that is the extracellular matrix, and this white thing here that is the fat. This is a moderate case where you can see that there are less muscle fibers, and we start to have a lot of this fat and fibrotic tissue. And this is an advanced case where you can see that there are only a few fibers, and almost all the muscle is replaced by this fibrotic and fat tissue. So how is fibrosis developed? As I said, it is very important the interaction that we are going to have between the muscle fibers that are injured, the inflammatory cells, these satellite cells, and the FABs. The problem is that in humans, we don't really know how these cells interact. There are not many studies that have been able to identify all the mechanisms or all the conversation between these different cells. What we do know is that when there is an injury in the muscle fibers or these inflammatory cells, they can release some factors, and some of them maybe are familiar to you, like for example TGF-beta or CTGF. And these factors will activate the FABs that can then proliferate, differentiate into fibrotic cells and produce the scar, the fibrotic tissue, or differentiate into adipocytes. But again, all these molecular pathways are not completely known. Something that is very relevant is that this process is not specific of Duchenne. It happens in all muscular dystrophies. So that's something that is, of course, very important, because if we have treatments that can reduce and control this process, this treatment can be effective not only for Duchenne, can be effective for many different diseases. Okay, so why is fibrofatty tissue bad for the muscle? For several reasons. First, because it does not contract, so it has no contractile properties, and this contributes to muscle weakness. Second, because it leads to muscle contractures and a stiffness. Third, because we know, and there are growing evidences of this, that the presence of fibrotic and fatty tissue in the muscle is not good for the muscle because it does not allow the muscle to regenerate properly, okay? It also perpetuates the muscle inflammation, and this means that the response to the drug can be worse if you have a lot of fibrotic and fat tissue. And also, it can also reduce the delivery of the drugs to the muscle fiber. So drugs aimed to reducing fibro-fatty tissue could be useful. And as I said before, not only for Duchenne, for many diseases. Okay, what can we do? From a strategic point of view, imagine that we want to design drugs that tackle fibrosis. So there are three options. One option is to block the production or the effect of these cytokines, of these TGF-beta and CTGF. Another option could be to reduce the capacity of the ability of the Fabs to differentiate. Another option could be to reduce the ability of the Fabs to proliferate. Let's start with the first. So there have been many treatments trying to block the TGF-beta and CTGF pathway. Some of them maybe are familiar with you. For example, losartan, it's an angiotensin II receptor inhibitor that is used in daily clinics and has demonstrated in some studies to be useful for the cardiomyopathy of patients with Duchenne. Other drugs like pirfenidone, for example, demonstrated to be useful in mice. As far as I know, it has not been tested in humans. A classic antifibrotic is alofungicin, and there was a clinical trial that was stopped due to adverse effects. And there are other multiple drugs. So if you read papers, you will see that there are many drugs that have been published trying to interact with these pathways. But there is a monoclonal antibody that I want to highlight that is pamrevlumab, which was tested in a phase 3 clinical trial. So this was an antibody that was interacting with this CTGF in a clinical trial that was called Lelantos. This may seem familiar to you. Some of you probably participated in these studies. And these were two studies that included non-ambulatory Duchenne patients and also ambulatory Duchenne patients. And unfortunately, these studies failed, did not demonstrate any effect of this drug. But this was the first time that one antifibrotic therapy, a monoclonal antibody, was tested in a large number of patients with Duchenne. So a second option, as I said, could be to reduce the differentiation potential of Fabs. And one good example of this could be givinostat. So some of you are already familiar with givinostat. Michela will present about givinostat this afternoon. And givinostat, it's a drug that inhibits this thing, the class 1 and 2 histone deacetylase. So what are histone deacetylase? So histone deacetylase are basically type of proteins that are going to open the boxes where the genes are. So we call that the chromosomes, and in general, the chromosomes needs to be open for the genes to be expressed. So when a cell needs to differentiate, imagine, into an adipocyte, into a cell that produces fat, it needs to produce a lot of these genes. It needs to open many of these boxes, right? So these drugs, what basically do is to close these boxes again. And this has demonstrated that can reduce the capacity or the ability of the Fabs to differentiate into fibrotic, into adipocytes. So the company that has produced this drug performed several studies in cells, and they show that the drug was able to produce big myotubes, which are like the big myofibers, when the cells were treated in vitro. And when they treated the animals, they also showed that there was a reduction in fibrosis. And I think that is very clear here that there was the muscle non-treated, and these were the muscles treated at different doses. So they improve the structure and the function of the mice. So in their clinical trial that is called EPIDIS, they have recently shown that the treatment showed a slow decline in some of the functional tasks for boys receiving givinostat, specifically an improvement in the Forrester-Klimb assessment. And as you know, this drug was approved by the FDA, and I think it is conditionally approved in the UK. Okay, I want to move to another type of a strategy. So we have talked about blocking the cytokines. We have talked about modifying the differentiation potential of the cells. I want to talk now about reducing the proliferation potential of the Fabs. And this is work that we have done in our lab. And basically, we have been using this drug that is called nintedanib. So nintedanib, it's a drug that is commonly used for pulmonary fibrosis. It's a drug that developed Boehringer Ingelheim many years ago and is now used for patients with this disease. So we were interested in nintedanib because blocks this thing here, what is called the platelet-derived growth factor receptor. And this is a receptor that all the Fabs express. So we were interested in this because all our Fab cells express this receptor. And what we show is that if we block this receptor with this drug, our cells, the Fabs, are not going to proliferate. So these are the cells that proliferate when they are treated with this cytokine. And when we treat them with the drug, they cannot proliferate. So they cannot expand the number. Moreover, we also observe that these drugs reduce the migration, so how the cells move in the muscle, and they also reduce a bit the expression of collagen, so the expression of this connective tissue. But the main thing that this drug was doing was to reduce proliferation. So we treated two different animal models. One was the MDX, which is the model of Duchenne, and the results were showing a mild decrease in the amount of collagen in the tissue. You can see here the decrease that we observed in the Western blot, but in the images was not very clear. And we decided to use a different animal model, which is much more fibrotic, which has a limb-girdle muscular dystrophy produced by mutations in this sarcoglycan gene. And here we could observe extensive reduction of the amount of fibrosis in the tissue with the drug. And very important, we also learned that this drug was in the tissue reducing the number of cells, reducing the number of Fab cells, and this was probably the effect of the drug. So all these drugs are showing that they can modify the function of Fab. So Fab seems to be important. And this is a protocol that we developed some years ago, and that has allowed us to do a lot of experiments in the lab. Because we can now isolate these Fabs from muscle samples of patients. So when we have a biopsy of a patient, what we do is that we process the biopsy in a plate. We basically mince the muscle. And at this stage, we can freeze the muscle and keep it in nitrogen tanks and culture many years after. When we culture the cells, when we culture these pieces of muscle, we can obtain cells. And using this machine that is called sorter, we can obtain our satellite cells. Remember, these are the ones that produce muscle, and the Fabs. These are the ones that produce fibrosis. And we can investigate these cells from humans in our labs. So that's the way how we use muscle biopsies from patients, okay? And in this project that has been funded by Duchenne UK, basically what we have done is to develop a protocol that allows to study the process of fibrosis and adipogenesis in the muscle. So we can culture our Fabs with TGF-beta, and this will make them to produce a lot of collagen, or we can culture the Fabs in a specific medium that will differentiate it into Fab, and then they will start producing this green thing here, which is the fat. And using this protocol, what we have basically done is to test a lot of drugs, what we call a high-throughput drug screening. So we have used human cells to test drugs that could have an effect in fibrosis and in adipogenesis. 500 different drugs in this protocol. And what we have seen are interesting things. So this is like the final graph, and whatever is on the right of this axis means that increases the amount of fibrosis and adipogenesis, is what we don't want. Whatever is on the left is reducing the production of fat and fibrosis. And this quarter here is when it's reducing at both high and low doses. So we identified that around 15 of the 500 drugs were able to reduce the amount of fibrosis, reduce the amount of fat that these cells were producing in the plates. And interestingly, this has allowed us to understand different molecular pathways that can be involved in this process. So we now know more about how this is controlled. And the next thing that we did is, this is all the list of the drugs that we tested. We confirmed these results in a large number of cells from controls. And because we also had muscle biopsies from patients with Duchenne, we also extracted the cells from Duchenne, and we confirmed these results in Duchenne and also tested that these drugs were not toxic. So this is the PhD of two of the PhD students, Priyanka and Zoe, that are working in the lab. And we are now entering in the last phase of the study where we are testing all these molecules in different animal models, in the murine models of Duchenne. And I expect that we will have results very soon to show you. So just some take-home messages before we end. So the first thing is fiber fat suspension is a common trait in muscular dystrophies. And as I show you, it has negative consequences in terms of muscle performance, regenerative capacity, and the delivery of drugs. So it's not good to have fibrosis and fat in the tissue. The molecular pathways in humans are not well known, so we need more research to understand these molecular pathways, because it's the only way that how we can develop drugs. Drugs with an antifibrotic effect have a positive impact, not only in fibrosis, but also they can improve muscle regeneration. And as you can imagine, we can combine drugs. So as I see, the future is in a combination of different therapies, using gene therapy with a combination of antifibrotic, pro-regenerative, and maybe anti-inflammatory drugs. So new antifibrotic drugs tackling the specific pathways involved in muscle fibrosis are needed, and this is something that we are doing research. And I think that human fabs are a good model to test all these drugs. And that's my last slide. I would like to thank especially this person here, who is Esther Fernandez-Simon, who is the postdoc working with me since now 10 years, and that has done most of the work that you have seen here. Thank you very much for your attention.